Here's a brief overview of the structure of DNA and comparing that to the structure of RNA. So first of all, it was Phoebus Aaron Levine who studied the chemistry of DNA for 40 years and discovered that DNA consists of a large number of linked repeating units that are called nucleotides. So nucleotide, which consists of a base, which can either be your purine or your pyrimidine, a sugar, which can either be oxyribose or deoxyribose, and then a phosphate group. Okay, so if we look at each one of these individually, first we will do the base. So we're going to get into the actual pyrimidines versus purines in the next video, but for this one I'm just going to draw a little rectangle as a base for both of these molecules. Okay, so this base will come down and attach to the sugar. And we'll draw the sugar in a different color. So let's pretend that this is RNA and this is DNA. So once again, this is deoxyribonucleic acid and this is ribonucleic acid. So if we look at the structure of the DNA first, we will see that it is a pentagon shape. So it is like this. Bad pentagon, but nonetheless. Okay. Pentagon. And each one of these carbons in this ribose sugar, so each point is a carbon. If you're taking organic chemistry, you'll see that every point in a cycloalkane is a carbon. So right here we have our 1' carbon. Let's do this in a different color. 1' carbon, 2' carbon, 3' carbon, 4' carbon, and then up here, which is attached to your, actually I should do this in red, attached to your phosphate group, is your 5' carbon. So in each one of these, so this is the ribonucleic acid, you're going to have your OH group up here that's attached to, so let's just say if we, there's your carbon, so this is a carbon, this is a carbon, and then down here you have in a ribonucleic acid, you have an OH group, the hydroxyl group, and then another hydroxyl group down here. And in the DNA, you have a hydrogen and a, oh, sorry, hold on. Oh, yeah, sorry, that's right. And a hydroxyl group. So obviously this is deoxy. You're removing an oxygen to make it a deoxyribonucleic acid. And the same thing here. We have our 5' carbon coming up here and connecting. And then on each one of these, we have a phosphate group. So we will draw the phosphate group now, which is attached to the carbon. And then we'll just do it as a circle. And attached to the carbon, and it's just a circle. And what a phosphate group is, if I just draw it down here, is a phosphate with an oxygen, an oxygen, an oxygen, and a double bonded oxygen. So obviously, if you took first year chemistry, you'll know that each one of these has a negative charge because it's only bonded to one thing, and the phosphate can't be bonded to anything else. So this has a negative charge. If this wasn't bonded, so if this was actually standing by itself, it would look like that. But since it is attached to this carbon, we remove this one, and it looks like that. So there you have it. It's basically the only difference between DNA and RNA is the addition of this oxygen, this hydroxyl group on the 2' carbon of the ribose sugar. And you will see in the next video that the base can also differ based on whether it is a DNA or RNA. DNA uses adenine, thymine, guanine, and cytosine, whereas RNA uses, instead of thymine, it uses uracil, which we'll discuss in the next video.